is being recorded. Assalamu alaikum. I, Dr. Maya Khan, on the behalf of organizers, welcome you all in the hybrid lecture entitled Polymer-Based Nanofibrous Membrane with Bioactive Zinc Oxide Nanoparticles for Advanced Wound Healing and Skin Regeneration, from Synthesis to Applications. This lecture is organized by Sindh Innovation Research and Education Network, SIREN, in collaboration with the International Center for Chemical and Biological Sciences, University of Karachi. Let me take an opportunity to introduce our respected speaker, Dr. GHULAM MURTAZA, who is a dedicated biotechnology researcher with specialization in nanobiotechnology and currently serving as a lecturer in Department of Molecular Biology and Genetics at Shahid Benazir Bhutto University, Shahid Benazir Awal. He holds a PhD in biotechnology in the Institute of Biotechnology and Genetic Engineering, University of St. John Shoro. He has several publications to his credit in national and international journals. His current research is at the intersection of nanobiotechnology and molecular biology, with specialized focus on developing advanced materials for regenerative medicine. So with no further ado, we welcome our respected speaker.
Respected focal persons, directors, research scholars, Assalamu alaikum. Today I am here to give a talk on the topic polymer-based nanofibrous membrane with bioactive zinc oxide nanoparticles for advanced wound healing and skin regeneration from synthesis to application. Before starting the session, I want to just reconnect your ideas, your mind from past to present research. So from past to present, just I want to connect your ideas as a scientific field to the science. So directly or indirectly, we all are belongs to science. So being a researcher, scientific researcher, in your opinion, what is science? How you can describe word science? Simply during my presentation, I may be asked some questions, just want your attention toward my side. So first question from you to you all, okay, what is science? Being a researcher, how you can describe the science? Without traditional definition, okay, science is a knowledge which we can get experiments and observations and so on. Beyond this, how you can describe the science? You define the science. Anyone?
Anything basically related to matter is science. Like anything, any knowledge, anything related to matter is science. Except spirituality, any other thing is
just only matter.
That's what is my personal definition.
Yes.
I would say it is to solve the problems of the mankind. We utilize our abilities.
Anyone else?
Science is a way of getting information.
Exactly. So being a researcher, in my opinion, science is nothing, it is just to explore the hidden secrets of nature. इसके अलावा साइंस और कुछ नहीं है. नेचर में जो कुछ प्रेजेंट है उसको एक्सप्लोर आउट करना, उसको फाइंड आउट करना, उसको यूटिलाइज करना इन डिफरेंट फॉर्म्स, दैट इज द साइंस. उसमें हम डिफरेंट टेक्निक्स अप्लाई करते हैं, डिफरेंट वेज अप्लाई करते हैं, उसके बेस पे हम जो वर्ड यूज करते हैं, दैट इज द साइंस. सो फ्रॉम पास्ट अगर हम कनेक्ट करें पास्ट टू प्रेजेंट, आज तक साइंस को कनेक्ट करें, कई साल पीछे चले जाएं, जब साइंस का कोई तस्वर नहीं था, उस टाइम क्या था? जस्ट जो है वो एक ह्यूमन बीइंग या ह्यूमन सोसाइटी जो है उसी में गुजारा करती थी कि जस्ट उसको जो है वो लाइक मैं एक वर्ड यूज करूँगा, लाइक एनिमल के, उसको जो है वो खाना मिल जाए, बस उसका गुजारा हो जाए, यही होता था. पर उसके बाद जैसे जैसे साइंस ने तरक्की की, उसके बाद क्या हुआ कि साइंस ने थ्रू साइंस जो है वो ह्यूमन ने डिफरेंट वेज जो है वो अडॉप्ट करना स्टार्ट किए फॉर देयर लिविंग, फॉर देयर लाइफ ऑफ स्टाइल. तो उसमें देखिए फर्स्ट ऑफ ऑल अगर जब इन्वेंशन हुई माइक्रोस्कोप की, माइक्रोस्कोप से पहले बियॉन्ड द आई साइट आईसाइट की जो लिमिट्स थी उसके बियॉन्ड किसी भी लाइफ की एग्जिस्टेंस नहीं थी। जैसे ही माइक्रोस्कोप की इन्वेंशन हुई उसके बाद सडनली जो है वो माइक्रोस्कोप की जो है वो इन्वेंशन से जो है वो माइक्रोस्कोप वर्ड का इन्वेंशन हो गया कि नहीं, कि ह्यूमन की आई के बियॉन्ड भी जो है वो लाइफ एग्जिस्ट करती है। दैट लाइफ इज कॉल्ड एज द माइक्रोस्कोपिक। means के through scientific nature we have to explore out another world that is the microscopic world. उसमें आपके microorganisms आ रहे हैं, bacteria, viruses, even के human body itself made from the microscopic objects. Cell itself microscope. Inside cell microscopic features like cellular organelles, nucleus. Inside nucleus again there is the microscopic objects, DNA, RNA. These all are the microscopic. So through science we have to explore out the word of the microscopic. Microscopic word को हमने explore out कर दिया. But today in the modern world, today in the scientific era of AI, era of modern science, we are going to inside the micro and nano world itself. हम खुद पहले हमने क्या किया, खुद जो नेचर में प्रेजेंट थी माइक्रो और नैनो वर्ल्ड उसको हमने फाइंड आउट कर लिया। उसके बाद अब nowadays we are going towards the nano and micro side. हम खुद जो है नैनो और माइक्रो ऑब्जेक्ट्स बनाने जा रहे हैं। That is the variation. So in my opinion, जो मैंने साइंस की डेफिनेशन बताई जस्ट, we have to explore the hidden secrets of life, nature. नेचर के जो हिडन सीक्रेट्स है उनको हम एक्सप्लोर आउट कर रहे हैं, फाइंड आउट कर रहे हैं। उसके हिसाब से हम उसमें जो है मॉडिफिकेशंस, रीमॉडलिंग करके न्यू जो भी जो है वो लाइफ के लिए बेनिफिशियल चीजें हैं वो हम जो है वो प्रोसेसेस कर रहे हैं, सिंथेसाइज कर रहे हैं। सिंपली हमने क्या कर दिया, वर्ड को नैनो कर दिया, कंसाइज कर दिया। So my topic is basically based on the synthesis of nanomaterials for wound regeneration. So let's start from the introduction of nanobiotechnology. What is nanobiotechnology? How you can utilize it? How we can use it? Simply, nanobiotechnology provides insights in the structural features of biological system such as cells, tissues, as well as to develop a nanobiomaterial for diagnostic, therapeutics, tissue regeneration. Nanobiotechnology is the development of fundamental materials, basic materials, devices, systems for understanding and control of the matter at nanoscale, nanoscale level, where new functionalities and properties of matter are observed and harnessed for a broad range of applications. It uses tools for nano or microfabrication for the synthesis of nanofibers to build a device for studying the biological systems. It gives exciting opportunities to produce or bring high impact advances in the field of bioengineering and medicines. So simply I have added here some applications of nanobiotechnology like drug delivery, gene therapy, food safety, various therapeutics, nanodiagnostic, tissue engineering. Our major focus is over on the tissue engineering. But inside tissue engineering is a broader field, but we just work on single aspect, that is the skin tissue engineering. Skin tissue engineering is the rapidly production of skin constructs that offer the complete regeneration of functional skins, including all layers of skin like epidermis, dermis, hypodermis, and all skin appendages like hair follicles, sweat glands, and sensory organs. So generally, skin is the largest organ of a body. Skin perform various functions, multiple functions. Like for example, we can say skin perform the function of immunity. How skin perform the function of immunity? How skin perform the function of immunity?
It is the first line of barrier against pathogens.
Exactly. Skin is the first line barrier to the entrance of micro or nano objects, even the dust particles. In this way, skin acts as the immunity. And then next step is the thermoregulation. Skin maintain the thermoregulation processes. How skin maintain the thermoregulation processes? By vasodilation, by vasocontraction. So skin maintain the thermoregulation processes. Similarly, skin is a sensory organ. Sensory organ किसको कहते हैं हम? Detecting any change within the environment. इन्वायरमेंट में जो चेंज आ रहा है उसको डिटेक्ट करना. हाउ इट कैन डिटेक्ट? बाय टचिंग, बाय हॉटनेस, टेम्परेचर की हॉटनेस, कोल्डनेस, ये सारी की सारी क्या है? फीलिंग्स हैं, सेंसरी ऑर्गन्स हैं. तो स्किन आल्सो परफॉर्म अ रोल ऑफ एज अ सेंसरी ऑर्गन. Similarly, a vitamin D metabolism. Due to the exposure of sunlight, skin carried out the vitamin D metabolism. Similarly, osmoregulation. Skin perform the osmoregulation by maintaining the osmotic pressure. If there is the excess of water, that will be excreted in the form of sweat with salts or other ions. Or if there is the shortage of water in body, it conserves the water. And the major aspect on which we are working, that is the self-regeneration of skin. Within cold season, अभी सर्दियों का सीजन शुरू हो रहा है, आपने देखा होगा कि आपकी स्किन जो है वो क्या हो जाती है? ड्राई हो जाती है. तो ये ड्राई स्किन क्या है? डेड सेल्स. कौन से डेड सेल्स? जो आपके अपर लेयर पे डेड सेल्स है वो है, पर उसमें से कभी भी आपको जो है वो बोन फील नहीं होता. क्यों नहीं होता? Due to the process of homeostasis, itself our skin regenerate and renew it. That is the normal process of homeostasis. But in case of deeper wounds, जब wound ज़्यादा हो जाए तो उस टाइम जो है वो homeostasis का process जो है वो affect हो जाता है. Then क्या होगा? In case of minor wounds, when a part of epidermis is damaged, the destroyed cells are then replaced by new ones from the basal layer of epidermis. However, if wound is the deep, the damage is more significant, making the healing processes more complex. Globally, over 6 million patients suffer from the severe injuries, among which more than 3 lakh persons die annually. Even though today acute burn care, rehabilitation and reconstruction remain a major problem worldwide. So skin wounds. Wound है क्या? Whenever there is the any layer of skin is damaged, there is the development of wound. वहाँ पे wound की development होगी. तो basically wounds की अगर हम बात करें तो 4 types के wound होते हैं. First one is the abrasion, second one is the laceration, third one is the avulsions, and fourth one is the penetrating wound. So if we talk about the abrasion wounds, abrasion is the type of superficial wound. When you fall on the rough surface, the upper superficial layer of skin is damaged. That is called as the abrasion wound. It's like in the figure. And second one is the laceration wound. Laceration wound is the jagged cut with sharp objects like knife. Knife के साथ कट लग जाए तो उसमें क्या होगा कि स्किन के जितने भी लेयर्स हैं वो आपके सारे के सारे क्या हो जाएंगे? डैमेज्ड हो जाएंगे. That is the incision with sharp objects. Another type, that is the avulsion wounds. Avulsion wounds separate the various layers of soft tissues so that they become either completely detached or hanged up. And the fourth one type is the penetrating wound. Penetrating wound with pointed objects, sharp objects. In हो जाए आपकी जो है वो skin में, that is called as the penetrating wounds. If there is the wound develop हो गया. So how one can repair? Wound repair किस तरह होता है? There are the different stages. Basically 4 stages for wound healing या wound repairing. First one is the homeostasis, second one is the inflammation, third one is the proliferation, and fourth one is the remodeling. So in homeostasis just maintain the balance of the body, stoppage of the bleeding. कि bleeding को क्या किया जाए? रोका जाए. Then inflammation में basically there is the role of WBCs. WBCs का रोल होता है जिसमें क्या होता है, जो inflammatory cells and nervous cells हैं उनको क्या किया जाए? Remove किया जाए. Proliferation में make a new skin tissues and repair broken blood vessels. That is the proliferation. And then last is the remodeling. Wound fully closed and the cells used to repair are no longer necessary. Then how healing starts? Healing किस तरह होती है? In some people, the healing process does not occur normally. In fact, in some ways, it may regenerate but create a scar. Scar लिखा होगा. After wound regeneration, there is the development of a scar. Scar की development होती है, वो reddish होता है, dark and brown color होता है, or the fair skin. These scars can also causing a itching. They are thick, hard to touch, and dark brown or reddish in color or the fair skin. In worst cases, outgrowths must be surgically removed. इतनी ज़्यादा हो जाती हैं कि हमें जो है वो सर्जरी भी करवानी पड़ती है उसको रिमूव करवाने के लिए, क्योंकि फेयर स्किन के ऊपर जो है वो एक किस्म का जो है वो एयर नज़र आ जाता है, जिसकी वजह से उसको हमें रिमूव करवाने की नीड आ जाती है. In this respect, particular wound dressings are needed that play a significant role in healing skin repair. Therefore, the ideal wounds are required. So, ideal wounds. Word here used ideal wound dressings. Ways of skin healings. Skin healings में वही बात अगेन आ रही है कि वुंड्स के बेसिस पे हीलिंग का प्रोसेस होता है. सुपरफिशियल वुंड है, एक्यूट वुंड है, या डीप या क्रोनिक वुंड है, तो हीलिंग का प्रोसेस भी जो है वो उसी हिसाब से होगा. हीलिंग अकर्ड थ्रू अ नेचुरल प्रोसेस में रिक्वायर अ मिनिमल लोकल थेरेपी. हीलिंग अकर्ड विदाउट स्कार फॉर्मेशन और मिनिमल स्कैरिंग. ठीक है, जो फर्स्ट टाइप था वुंड का, उसमें क्या होगा, नॉर्मल जो सुपरफिशियल लेयर स्किन की डैमेज हुई थी वो रिपेयर हो जाएगी, बट देयर इज नो डेवलपमेंट ऑफ स्कार. उसमें कोई भी स्कार की डेवलपमेंट नहीं होती. Another type, that is the acute wound or cuts, required continuous assistance management like suturing of wounds over alternate days, different types of wound dressings. Healing process is slow. Normal tissues are replaced by fibrous tissues like keloid formation. Large or extensive wounds. For large cutaneous wounds, the better results obtained by synthetic wound scaffolds. These are artificially synthesized, especially focus on the synthetic skin scaffolds, which is designed by the process of nanotechnology to get a better results and quick recovery with a less morbidity and mortality rate. So if we talk about the ideal dressing, wound dressing, so what are the characteristics of ideal wound dressings? Particularly, wound dressings needed to play a significant role in wound healing and wound skin repair. Mechanical stable होगी, entirely suitable architecture होगा, biodegradable होगी, biocompatible होगी wound dressings. Protect from the bacterial contamination and reduce formation of scar. Fourth one is the be comfortable, sterile, non-allergic, non-adherent, and non-toxic. Regulate humidity, excrete wound exudates, permit thermal insulation, mechanical protection, allow gases exchange. These all are the characteristics of an ideal wound dressings. So what are the pharmacological needs for such type of ideal wound dressings? The pharmacological needs of modern regenerative medicines is to design effective nanofibrous wound dressings material that perfectly exhibits a wound healing, antibacterial activity, anti-inflammatory properties, further have ability to release desired drugs in a controlled and targeted fashions. These are the pharmacological needs based on the ideal wound dressings. Characteristics like antibacterial भी होगी, antifungal भी होगी, और release property भी होगी. Release property for what? For the loaded bioactive molecules. जो उसमें loaded bioactive molecules होंगे उसके लिए. So how these pharmacological needs completed? These pharmacological needs are completed by the development of nanocarriers. Nanocarriers basically in two forms. First one is the nanofibers and another is the nanoparticles. Carrier word, where we should use carrier. Carrier word carries something from one place to another side or targeted side. एक जगह से दूसरी जगह या सेल्स की तरफ जो है वो किसी चीज को पहुंचाना है, तो उसके लिए हम जो यूज करते हैं वो टारगेटेड मॉलिक्यूल्स जो है वो यूज करते हैं. तो platform where the drug bioactive molecules could be introduced inside the polymers, carry them towards the targeted regions. Nanoparticles is an interesting medicine. Out of them, one is the nanocarrier. First one is the nanoparticle. Limitations of conventional drugs in human body, including drug resistance, lack of selectivity, lack of water solubility, was observed. Recently, scientists are going to find out proper ways to replace old and traditional medicines by novel materials, such as effective nanoparticles, nanofibers, special nanofluids as a drug. Nanoparticles have high surface area to volume ratio. This allows for many functional groups to attach to a nanoparticles so it can bind to specific cells. Nanofibers have active properties against microorganisms such as antibacterial as well as having excellent antifungal activity. Another type of nanocarrier is the nanofibers. Skin regeneration through the scaffolds in the form of nanofibers. Tissue engineering scaffolds must promote the cell growth, maintain physiological functions, and establish the normal state of the cell differentiation. Nanofibers are particularly interesting when used as a wound dressings material and scaffolds for tissue engineering because of its good biocompatibility, cell adhesion, water permeability, biodegradability, minimal inflammatory reactions. The initial cell attachment and spreading of cells over the scaffolds is the important factor in the development of wound dressings. So numerous research have been developed till date. From these reports, nanofibrous reports have indicated that electrospin nanofibers are the best and useful in wound dressings material as compared to other like hydrocolloids, hydrogels. The best one is the nanofibrous loaded, nanofibrous based biomaterials. Tissue engineering scaffolds are nanofibers. With advanced in the field of tissue engineering, it is increasingly recognized that biodegradable, biocompatible scaffolds incorporated with multiple wound healing mediators, these are the carriers, might serve as the most promising medical devices for skin tissue regeneration. The nano dimensions of the nanofibers naturally gives it high surface to volume ratio, which lead it to enhance cell proliferation, migration, differentiation, high porosity, help in oxygen exchange and nutrient transport. Scaffolds typically made from the polymer biomaterials. How these scaffolds are made? Basically, these scaffolds are made from the biomaterials. Provide the structural support for cell attachment and subsequent tissue development. Scaffolding development. Which type of biomaterials we are using for the formation and synthesis of these type of scaffolds. So we can use different types of biomaterials, natural as well as synthetic, and blends of both, natural as well as synthetic. Natural polymers like collagen, cellulose, chitosan, gelatin, elastin, even DNA, RNA, that is also polymers. And synthetic polymers like PLA, PLCl, PLGA, these are other types of synthetic polymers. And third category, that is the blend. Blend में क्या होगा? Mixing of both natural as well as synthetic polymers. In our study, we have used a blended polymers. We have mixed a natural as well as synthetic polymers. So how such type of nanofibers are synthesized or formed? A very simple technique is used that is called as the electrospinning. Anyone have idea about electrospinning?
Yes.
So what is it?
It is a machine consists of many parts. You have two electric cars with high voltage, and
Exactly.
you have steering. You put your liquid
Solution.
inside the steering, and the steering you have another rope connected to the cathode, and covered with connected. It is like aluminum foil.
That is correct.
Using injection using some pumps, and under the field of high electric voltage, this solution can
In the form of fibers connected over the collector.
Yeah.
Yes, exactly.
It's not available here in Ireland.
Exactly. Electrospinning is a very simple technique used for the synthesis of nanofibers. Just solution converted into the fibers form, nanofibrous form, collected over the maybe aligned or random. When we come on the next slide, we will discuss about how random fibers are collected, how aligned fibers are collected. So this is the simple electrospinning setup. simple electrospinning setup is basically based on the four major components. First one is the syringe pump, needle, voltage supplier, and the collector. These four components are the important in electrospinning. Syringe pump, this is the syringe pump, which controls the flow rate of the solution. At what rate we want to flow the solution, either 1 ml per hour, 0.5 ml per hour. The rate will be adjusted according to the synthesis of nanofibers. Nanofibers जिस तरह हमारे बनेंगे, जिस रेट पे बनेंगे उसी रेट को हम जो है वो एडजस्ट करेंगे फ्लो रेट को. Then second one is the needle. This is the needle, that is the metallic needle, which shifts the solution into the electric field. Electric field will be provided by the voltage supplier. This voltage supplier provide a current, high voltage, up to 15, 80, 20 kV तक करंट देता है. इसका पॉजिटिव पोल जो है वो हम अटैच करते हैं विद द नीडल एंड नेगेटिव पोल इज अटैच्ड टू द कलेक्टर where the nanofibers are attached. As the current is provided, the drop of solution, ejected drop from the needle, then starts converted into the cone. Like pulling, जिस तरह pull pressure होता है, pulling pressure होता है, क्योंकि ये पॉजिटिव और नेगेटिव में जो है वो क्या क्रिएट होगा, अट्रैक्शन. नेगेटिव और पॉजिटिव पोल्स अट्रैक्ट करते हैं, तो अट्रैक्शन की वजह से जो ड्रॉप ऑफ कोन होगा सॉल्यूशन का, उसमें एक पुलिंग फोर्स क्रिएट होगा. उस पुलिंग फोर्स की वजह से ड्रॉप ऑफ सॉल्यूशन कन्वर्टेड इनटू द फाइब्रस फॉर्म, nanofibrous form. That fibers are started to collect over the collector. Collector may be the plain sheet, may be the rotating membrane. If we just need a random fibers like in this image, we use plain collectors in the form of aluminum foil, in the form of metallic sheets. But if we have need of aligned fibers, straight fibers, we use our rotating membranes, on which aligned or straight fibers are collected. So simply it is the machine. So different parameters are also affecting over it. Solution parameters भी होंगे और environmental parameters भी होंगे जो इलेक्ट्रोस्पिनिंग के ऊपर इफेक्ट कर रहे होते हैं. सॉल्यूशन पैरामीटर्स में जो है वो स्पेशली पॉलीमर मॉलिक्यूलर वेट है, पॉलीमर की कंसंट्रेशन, विस्कोसिटी, इलास्टिसिटी, कंडक्टिविटी एंड सरफेस टेंशन. These all are the factors affect over the synthesis of nanofibers. If our solution is not properly synthesized, if our solution have not proper concentration, nanofibers never form. So first must you have to prepare a proper solution with proper concentration. If there is no proper concentration, no proper synthesis of nanofibers. There is the development of beads, there is the development of short spots like droplets. Environmental factors like temperature and humidity also have a major factor during the electrospinning processes. So aim and objectives. Aim always एक ही होता है, उसके बेस पे जो है वो आपके objectives सेट होते हैं. Like for example आप PhD के स्कॉलर्स हैं, तो आपका एम क्या है कि PhD की डिग्री करना है. उस PhD की डिग्री पे बेस पे आप डिफरेंट ऑब्जेक्टिव सेट करते हो. आपको GRE भी देनी है, आपको कोर्स वर्क भी करना है, आपको रिसर्च वर्क भी करना है, आपका जो है वो पेपर पब्लिकेशन भी होना है. These all are the objectives to achieve just one aim, that is the PhD. ठीक है. Similarly in research, even though you all are the researchers, in PhD you just work on a single topic, just simple topic. एक टॉपिक होता है, उस टॉपिक के बेस के ऊपर जो है वो मल्टीपल पैरामीटर्स होते हैं. These are called as the objectives. ठीक है. तो एम हमारा क्या है, नैनोफाइब्रस बेस्ड एक स्कैफोल्ड बनाना है जो वो डीलिंग में क्या हो, हेल्पफुल हो. उसके बेस पे जो है वो इतने सारे ऑब्जेक्टिव्स हैं जिनको हमें अचीव करना है. ऑब्जेक्टिव्स में फर्स्ट हम जो है वो बायोएक्टिव नैनोफाइब्रस स्कैफोल्ड्स बनाएंगे बाय यूजिंग सिंथेटिक और नेचुरल पॉलीमर ब्लेंड. मैंने बताया था दोनों का. PLCL that is the synthetic polymer and chitosan that is the natural polymer. Then the synthesis of zinc oxide nanoparticles by two different methods, chemical synthesis as well as the green synthesis. Blending of these bioactive molecules into the nanofibers. Then characterization. जब हम कोई मटेरियल स्पेशली ह्यूमन बीइंग के लिए इन द फॉर्म ऑफ मेडिसिन जब बना रहे होते हैं तो उसकी दोनों तरह की प्रॉपर्टीज होती हैं, बायोलॉजिकल कैरेक्टराइजेशन एज वेल एज फिजियोकेमिकल कैरेक्टराइजेशन. जब तक हम दोनों नहीं करते तब तक हम उस मटेरियल को अप्लाई नहीं कर सकते ओवर द ह्यूमन हेल्थ। तो फिजियोकेमिकल कैरेक्टराइजेशन में ये होता है कि मटेरियल की जस्ट हम कैरेक्टराइजेशन करें कि अगर हमारा मटेरियल जो है वो प्रॉपर बना है कि नहीं बना। उसके बाद बायोलॉजिकल कैरेक्टराइजेशन में ये होता है कि आवर मटेरियल इज इफेक्टिव और द बायोलॉजिकल साइड और नॉट। उसमें एंटीबैक्टीरियल एक्टिविटी, एंटी फंगल एक्टिविटी, एंटी कैंसरस एक्टिविटी, साइटोटॉक्सिसिटी और एनिमल्स के ऊपर जो है वो हम उसके फाइनली लास्ट में जाकर ट्रायल्स करते हैं। Simply, this is the graphical abstract. And graphical abstract, first step is the synthesis of the nanoparticles by two methods, chemical as well as green synthesis. And second one, there is the development or synthesis of polymeric solutions. From these polymeric solutions, there is the development of nanofibrous membranes through the process of electrospinning. Loading of zinc oxide nanoparticles as well as nickel-amide into the nanofibers. Then starts characterization. First one is the physiochemical characterization. For morphological analysis, SAM should be performed. FTIR, XRD, UV visible spectroscopy, zeta potential, release profile should be performed. Then in vitro testing like cytotoxicity, antibacterial activity, antifungal activity should be performed. In the last, in vivo testings for the animals, especially rats. So these are the materials and instruments or animals required during the study. This is the nicotinamide as well as tryptophan. इसकी properties हैं कि क्या है इसमें। Then हम आते हैं synthesis of zinc oxide nanoparticles. How we can synthesize the zinc oxide nanoparticles? Zinc oxide nanoparticles in our research, there is a lot of methods, but we have just follow a simple method, that is the sol gel method. Dr. Jeffrey Brinker is the pioneer of this method. Synthesis of nanomaterial through the sol gel method and set an important guidelines for sol gel. A conference named International Sol Gel Conference, which is held every 2 years at Petersburg, Russia. The sol gel process is more chemical method for the synthesis of various nanostructures, especially metal oxide nanoparticles. These are some properties of zinc oxide nanoparticles. So how synthesis starts? Synthesis starts initially with the 20 grams of zinc acetate dihydrate dissolved in 150 ml of distilled water. Solution was stirred for 20 minutes using magnetic stirrer until a clear solution turbidity was obtained. 80 grams of sodium hydroxide dissolved in 100 ml of distilled water, stirred at room temperature, then produced a sodium hydroxide solution. Slowly and gradually mix both the solutions. At the end, titration reaction was performed by the addition of 100 ml ethanol in dropwise manner on continuous stirring. Centrifuge at 3000 rpm for 30 minutes. Dried at a high temperature, 80 degrees centigrade for 5 hours. In the last, calcination process should perform within the furnace at 400 degrees centigrade for 5 hours. Then in the next step, there is the synthesis of zinc oxide nanoparticles by green synthesized method. In this method, we have used Ocimum basilicum leaves for the synthesis of zinc oxide. First, we have to collect the fresh leaves of Ocimum basilicum, dry the leaves, grind it, make the powder. 20 grams of powder dissolved in 100 ml of deionized water. Heat it for 15 minutes, filtration, then centrifugation process at 3000 rpm to collect the filtrate for 30 minutes. In the next step, 5 grams of zinc acetate dihydrate dissolved in 100 ml of deionized water, stirred at 80 degrees centigrade for 20 minutes. Zinc acetate solution to extract added dropwise manner. Then the color of solution spontaneously changes. Zinc oxide nanoparticles precipitates forward and color starts to change towards the yellowish. Mixture was heated at 80 degrees centigrade for 2 hours. Calcination at 400 degrees centigrade for 5 hours in the furnace. Finally, a nanoparticle powder is produced, which is collected for further characterization. Now starts the synthesis of polymeric solution. Up to that, we have synthesized just nanoparticles. Now we are going to synthesize nanofibers through the process of electrospinning. Before electrospinning, we must have to prepare the solutions. So we have 6 different types of solutions with different bioactive molecules. Okay. Then electrospinning setup. How we have to run? Run these solutions separately. Each solution run individually. These are the parameters set during the electrospinning process. Syringe size, that is the 10 ml syringe, was used. Needle diameter 21 gauge. Voltage जो provide किए गए थे during the study, during the process of electrospinning, that is the 12 kV kilovolt. Distance between जो distance set किया गया needle और collector के दरमियान, that is the 15 जो है वो centimeters. Simply जो है वो ये diagram में आप देख सकते हैं. This is the syringe pump. Here is the fixation of syringe, which controls the flow rate. Here flow rate is adjusted at 1.5 ml per hour. Here is the needle attached with the positive pole of the current supplier, and this is the collector on which fibers are collected. How these fibers are separated from the collector? Basically, the fibers fall on the collector, they are attached with the collector. So first we have to specify a particular position on collector where there is the high concentration of nanofibers are present. After specification of position, then with the help of tweezers, slowly and gradually we have to remove the membrane from the collector. Then we have to pack it into the airtight bags for further characterization, either physiochemical characterization or biological characterization. Material must be first physiochemical characterization performed, because first we have to confirm that our material is successfully synthesized or not. Then we are going for further biological testings. So in physiochemical testings, first one is the morphological analysis of nano material in the form of nanoparticles as well as nanofibers. For morphological analysis, same scanning electron microscopy should be performed. या 10 जो है वो हम परफॉर्म करते हैं. तो बेसिकली हमने जो परफॉर्म किया है that is the same. तो सिंपली इंस्ट्रूमेंट जो यूज करेंगे, ये मॉडल है इंस्ट्रूमेंट का. किस तरह करते हैं हम? बेसिकली जब भी हम किसी प्रोसेस के लिए जाएं तो उसका हमें थोड़ा बहुत मैकेनिज्म जरूर आना चाहिए कि हाउ दैट मशीन वर्क्स. तो वो मशीन किस तरह वर्क करती है, किस तरह जो है हमें पर्टिकुलर मटेरियल की कैरेक्टराइजेशन करके दे रही है. तो फर्स्ट वन हमने जो मेम्ब्रेन बनाई, दैट इज द इन द फॉर्म ऑफ नैनो फाइबर्स. जो मेम्ब्रेन थी या इन द फॉर्म ऑफ पाउडर जो नैनो पार्टिकल्स थे हमारे पास, वो नॉन कंडक्टिव थे. तो इलेक्ट्रो जो सेम है वो हमारी वो जो है वो ऐसे समझें कि जो नॉन कंडक्टिंग ऑब्जेक्ट्स हैं उनको रीड नहीं कर पाते. तो फर्स्ट हमें जो है उनको क्या बनाना है, मेटल बनाना है, कंडक्टिव बनाना है, ताकि इलेक्ट्रिक करंट जो है वो इजीली उनमें से पास हो सके. तो हाउ वी कैन मेक देम बाय कोटिंग विथ गोल्ड. Dried samples of nanoparticles or nanofibers were sprouted or coated with gold before scanning using 10 kV accelerating voltage and taken out the images. Average fiber diameter was measured using images software. उसके थ्रू जो है वो average fiber diameter हम measure करेंगे, that is the software, images software. And then finally, results are present in the histogram chart that's made within the origin software. Then FTIR. FTIR performed for the chemical characterization. Same for morphological analysis, our material have proper morphology, proper shape or not. And then for chemical characterization, there must be the FTIR. Fourier transform infrared spectroscopy for chemical group analyzation of the material, nanofibers as well as nanoparticles. Instrument model, this is the model. Resemblance between all of the prepared samples were checked against zinc oxide nanoparticles. Finally, results are present in the stacked line chart. Then XRD, X-ray diffraction. XRD used to analyze the crystalline nature of the material. Can our material have proper crystalline geometry or not? By using an instrument, this is the model of the instrument, Bruker's X-ray diffractometer. X-ray diffractometer basically consists of three basic elements: X-ray tube, sample holder, and an X-ray detector. From X-ray tube, there is the ejection generation of the X-rays, and sample holder which holds the sample. As the X-rays are released or ejected from the X-ray tube, they passes from the sample. There is the production of X-ray spectra. X-ray spectra are detected by the X-ray detector, which converts it into the peaks or in the numbers. Then we are going to biological characterization. In biological characterization, first we perform in vitro biological characterization, then in vivo characterization. If we obtain successful results in vitro, then we go towards the in vivo. In vitro testings, first there is the performance of antibacterial activity. Antibacterial activity performed by the zone inhibition method, in which the growth media is used, LB growth media. This is the composition of LB growth media in 1 liter. Autoclaved the media at 121 for 15 minutes. Poured it into the petri dishes under sterilized conditions using laminar airflow cabinet. Wait for 5 minutes to 10 minutes just for solidification of media. After the solidification, inoculate the bacterial culture through the streaking method from freshly activated culture. These three types of cultures we have used: E. coli, Streptococcus, Staphylococcus aureus. Inserted the discs over the bacterial culture and made the replicates. Press the discs down with steel saw to make contact with the surface. You do not want to smash it over the agar itself. Place your plate agar side up inverted position at 37 degrees centigrade within the incubator. Examine the plates after 24 hours. Measure only the zones showing the complete inhibition by visual inspection. अगर वहाँ पे जोन्स बन रहे हैं तो आप उनको मेजर करें, अगर जोन्स नहीं बने तो या तो एक्टिविटी अगेन रिपीट करें, हो सकता है आपकी हैंडलिंग एरर हो. अगर अगेन रिपीटेशन के बाद भी आपकी एक्टिविटी नहीं आ रही, मीन्स के योर मटेरियल विल नॉट possess antibacterial activity. Hold the measuring device, that is the vernier caliper, or the back of inverted plate. Place on the non-reflective surface. Compare the obtained values to determine the susceptibility level of used material against the microorganisms. Then antioxidant activity. Antioxidant activity evaluate on the basis of radical scavenging effect by using spectrophotometric method, determined by using DPPH assay according to the Chong et al. Principle क्या है इसका? Principle ये है कि जो एंटीऑक्सीडेंट कंपाउंड्स होते हैं वो एनिलेट करते हैं फ्री रेडिकल्स को, उनके इफेक्ट्स को क्या करते हैं, रिड्यूस कर देते हैं. एनिलेट द DPPH radical, itself DPPH is a free radical. So how we can perform this? First we have to prepare the DPPH solution. 0.1 mm. 0.1 mm DPPH solution was prepared by dissolving 3.94 milligrams of DPPH in 100 ml of methanol. Then we prepared the extracts of our samples, nanoparticles as well as plant extract. क्यों प्लांट एक्सट्रैक्ट का क्यों यूज कर रहे हैं? क्योंकि जो नैनो पार्टिकल्स हमने बनाया है वो उसी प्लांट एक्सट्रैक्ट से बनाए हैं. तो चेक करें हम कि आया प्लांट एक्सट्रैक्ट में ज्यादा एंटीऑक्सीडेंट है या जो हमने उससे नैनो पार्टिकल्स बनाए हैं उनकी एक्टिविटी जो है वो ज्यादा है. इसलिए फॉर चेकिंग पर्पस प्लांट एक्सट्रैक्ट इज आल्सो यूज्ड. सो प्रोसीजर में क्या होगा? 4.9 ml ऑफ मिथेनोलिक DPPH सॉल्यूशन प्लस 0.1 ml ऑफ सिंपल सॉल्यूशन ऑफ लीफ एक्सट्रैक्ट, जिंक ऑक्साइड नैनो पार्टिकल्स, chemically synthesized, green synthesized in separate bottles. Reaction mixture was incubated in a dark conditions at room temperature for 30 minutes. After that, deep violet color, as we put them in a dark, the color of solution is changed to pale yellow in the presence of zinc oxide nanoparticles or plant extract. The absorbance were taken through the UV visible spectrophotometer at 516 nanometers, depicts the decrease in absorbance as compared to control. रेडिकल स्कैवेंजिंग एक्टिविटी हम किस तरह कैलकुलेट करेंगे, मेजर करेंगे उसकी परसेंटेज. by this formula. Absorbance control minus absorbance sample divided by absorbance control into 100. Then the release, जो sustained release rate है वो हम किस तरह measure करेंगे? निकोटिनामाइड का रिलीज रेट के लिए हम जो है वो आर्टिफिशियल एक रिलीज मीडियम बनाते हैं. दैट आर्टिफिशियल रिलीज मीडियम इज कॉल्ड एज द फॉस्फेट बफर सेलाइन पीबीएस. सो इंस्ट्रूमेंट जो इसमें यूज करेंगे हम स्पेक्ट्रोफोटोमीटर. वेवलेंथ कौन सी वेवलेंथ पे रीडिंग लेंगे? 289 नैनोमीटर्स पे फॉस्फेट बफर सेलाइन होगा. ये कंपोजिशन है 1 लीटर फॉस्फेट बफर सेलाइन की. Place UV treated nanofibrous membrane of 4 by 4 centimeters and 10 ml of PBS. After a specific interval, 4 ml of liquid from the sample, जिसमें हमने sample डाला है, वहाँ से हम जो है वो 4 ml निकालेंगे into a separate bottle and replace with fresh PBS. Taken out, ये repeat करते रहेंगे हम जो है वो up to 96 hours तक. स्पेक्ट्रोफोटोमीटर रीडिंग्स जो है वो सैंपल्स की लेंगे हम 289 मीटर्स पे. देन एब्जॉर्बेंस जो है वो कन्वर्टेड रिलीज परसेंटेज बाय यूजिंग फार्मूला. ये फार्मूला यूज करके हम रिलीज परसेंटेज फाइंड करेंगे कि किस रेट पे जो है वो ड्रग रिलीज हो रही है नैनो फाइबर से. रिलीज ऑफ निकोटिनामाइड डिवाइडेड बाय टोटल लोडेड निकोटिनामाइड इनटू 100. देन द साइटोटॉक्सिसिटी टेस्ट. Cytotoxicity test was performed at Ekikhan Institute of Biotechnology and Genetic Engineering, University of Karachi. Cytotoxicity test was performed by MTTSSA. How can performed? Made a discs of 10 mm. Short disc बनाएंगे हम 10 mm size की nanofibrous membranes की. उनको हम sterilize करेंगे under UV light for 4 hours. Then हम क्या करेंगे? डिस्क सोल्व कर देंगे विद इन द पीबीएस सॉल्यूशन एट pH 7.4. इन द नेक्स्ट स्टेप NIH3T3 माउस फाइब्रोब्लास्ट सेल लाइन्स जो है उनको यूज किया जाएगा फॉर साइटोटॉक्सिसिटी टेस्ट एंड इनोक्यूलेटेड इन 96 well plate. Different concentration of samples were added in a separate wells. After incubation, 20 microliters of MTSSA was added in each well. The plate was interpreted through a laser plate reader at the wavelength of 290 nanometers. Cell viability after the exposure of nanofibrous membranes can be calculated by formula given value, that is the absorbance of sample divided by absorbance of length into 100. Then in vivo assessments. After the successful results of in vitro, then we are going towards the in vivo. In vivo, we have used rates. Rates were purchased from your HEJ Animal Health Research Institute of Chemistry, Karachi, Pakistan. First, we have anesthetized the rates by using the intraperitoneal injection of 5% ketamine and 2% xylene syringe. After anesthesia, hair removing over the particular site where we want to create a wound in red. After hair removing, antiseptic particular site on the skin. Then with the help of biopsy punch, we are going to create a wound, or we are going to develop a wound over the skin of the legs with the help of this biopsy punch. After creation or induction of a wound, we are going to stitch the synthesized nanofibrous membrane. Synthesized nanofibrous membrane stitch over the wound with surgical needle and thread. Stitching can be done when covered with a plaster and transfer them to the cage for a particular time period. 3rd day, 6th day, 9th day and 12th day. After particular days, we have to take out each rate and work rate from each group and observe their characteristics of regeneration. Examination of regeneration after the treatment with nanofibrous membranes. So on 3rd, 6th, 9th and 12th day, these are the days set for each group of treatment wound were examined. In such a way, on particular day, one rate of each group was anesthetized, open their bandage and measure the wound size with digital vernier caliper. Okay, how much wound recovery. Take the photo with a digital camera of regenerated wound of each rate. Size of regenerated wound was calculated and presented in the graph. Then microscopic observations for biopsy. Biopsy performed at DR lab of lungs, Jamshoro, Hyderabad. Most widely used staining technique, that is the H&E staining technique used. Tissues from regenerated wounds were collected and stored at 10% formaldehyde. After 6 hours of fixation, split the tissues in half with razor, that is the splitting or cutting. The tissues then undergoes for further processing like dehydration, staining, clearing, infiltration, and mounting. These are the steps of slide preparation for biopsy. First, the collection of tissues, in formaldehyde, fixation, remaining tissue processing, embedding, sectioning, staining, and then slide preparation, and finally observe under the microscope. Results, first results, we have present here the zinc oxide nanoparticles of both chemical as well as green synthesized. These are the results of chemically synthesized nanoparticles. These are the results of green synthesized nanoparticles. Here these are the results of nanofibrous membranes. Different nanofibrous membranes. We have made 6 different solutions. So we have prepared 6 different nanofibrous membranes. PLCL chitosan, this is the control in which no nanoparticle or no drug was loaded, and another here starts loading of bioactive molecules one by one. First, at initial stage, we have loaded the nicotinamide, and in histograms we have to measure the nanofibrous diameter size in each diagram. Okay, how much nanofibrous diameter is presented. In control, we have produced a 210 nanofibrous diameter, and in nicotinamide loaded sample, this is the 160 nanometer fibrous diameter. This is the chemically synthesized zinc oxide loaded nanofibrous membrane. Here produced the fiber diameter size, that is the 192.0 nanometers, and in nicotinamide as well as chemically synthesized nanoparticles loaded nanofibrous membrane produced 187.0 nanometers fibrous diameter. BD structures shows that there is the successful loading of chemically synthesized nanoparticles. Here is the green synthesized zinc oxide nanoparticles loaded into the nanofibrous membranes. In this histogram, only the green synthesized nanoparticles loaded into the nanofibrous membrane produced a fiber diameter, that is the 95.5 nanometer. In the next, here is the green synthesized nanoparticles as well as nicotinamide loaded nanofibrous membranes produced 133.4 nanometers. There is the variation of the diameter size in each membrane. This variation is why? This variation is due to the variation in the concentration of solution. As the concentration of solution is varied, the polymer size, the diameter size is varied. Chemical characterization. Different membranes were characterized for the FTIR. This is the pure chemically synthesized zinc oxide nanoparticles whose major peak at 430 and stretching peak lies at 630, and nicotinamide's peak at 3155, 150 and 1028. So these two peaks are successfully shifted into the nicotinamide as well as chemically synthesized zinc oxide loaded nanofibrous membranes. Again, there is the loading of green synthesized nanoparticles with nicotinamide into the nanofibrous membrane. Their peaks lies at, especially the green synthesized nanoparticles peak lies at 470 and 616, that is the stretching peak, and nicotinamide peaks are successfully shifted or present into the green synthesized loaded nanoparticles as well as nicotinamide loaded membrane. Then the UV visible spectroscopy. UV visible spectroscopy, synthesis of nanoparticles were confirmed by UV visible spectroscopy, simple by dispersing them into the deionized water. Data are used to calculate the energy band gap by using formula. Zinc oxide chemically synthesized having a strong peak lies at 365, whose optical band gap measured by the formula, that is the 3.35 electron volt. While the green synthesized zinc oxide nanoparticles strong peak lies at the 370 nanometers, whose optical band gap is measured as the 3.39 electron volt. Then the zeta potential of the synthesized nanoparticles chemically as well as green synthesized dynamic light scattering technique, that is the DLS, used to examine the zeta potential of nanoparticles. Zeta potential measured minus 17.1 millivolt in zinc oxide nanoparticles chemically synthesized, while minus 23.3 millivolt in green synthesized nanoparticles. Negative surface charge is owning the binding capacity of compounds to the extract with nanoparticles. X-ray diffraction, these are the peaks obtained during the X-rays diffraction in green synthesized ZNO as well as in chemically synthesized ZNO. X-ray diffraction pattern of ZNO nanoparticles consists of series of peaks with various intensities. Spectrum shows the peaks versus measured diffraction pattern at 2 theta. These are the angles on which the results obtained. Peaks, these obtained peaks in chemically synthesized nanoparticles, and these peaks are obtained in the green synthesized nanoparticles. After that, antibacterial activity results. Antibacterial activity carried out by zone inhibition method, fresh culture of these 3 types of bacteria, E. coli, Streptococcus species, Staphylococcus aureus, isolated from the wounds especially, at the diagnostic and research lab of Jamshoro. Results were checked after 24 hours of incubation. Zone of inhibition was measured with a digital vernier caliper, while area of inhibition was calculated by using this formula, pi d square upon 4. These are the results of just nanoparticles, not nanofibrous membrane. Chemically synthesized nanoparticles, zinc oxide nanoparticles, against E. coli, Streptococcus species, and Staphylococcus aureus, measured their zone of inhibitions. This is the green synthesized zinc oxide nanoparticles antibacterial activity against E. coli, Streptococcus species, Staphylococcus aureus, and their measured zone of inhibitions, and presented them in the chart. The highest inhibition activity were achieved achieved by green synthesized zinc oxide nanoparticle against Staphylococcus aureus, that is the 13.7 nanometers, and the area of inhibition, that is the 133.3 mm square. This is the antibacterial activity by nanofibrous membranes. Now nanofibrous membranes. But first nanofibrous membrane, that is the loaded with zinc oxide nanoparticles. कौन से nanoparticles? तो chemically synthesized nanoparticles हैं. उनके against. तो E. coli के against zones प्रोड्यूस हुए Staphylococcus aureus के और Staphylococcus aureus के, और उनके zones जो हैं वो measure किए गए. अगेन जो है वो Staphylococcus aureus के अगेंस्ट जो है वो हमें हाईएस्ट जो है वो इनहिबिशन जोन मिला है बाय दिस मेम्ब्रेन PLCL chitosan, जिसमें nicotinamide as well as nanoparticles loaded nanofibrous membrane produced the highest zone, that is the 14.0 plus minus 3, and the area of inhibition, that is the 135.8 mm square. Now again, antibacterial activity. This is the antibacterial activity of nanofibrous membrane loaded with green synthesized nanoparticles against E. coli, Streptococcus, Staphylococcus aureus, and their measured zones. Again, here we obtained a highest zone against Staphylococcus aureus by this membrane loaded with green synthesized membrane, that is the 14.9, and the area of inhibition, that is the 174.2 mm square. From overall antibacterial activity, the highest activity of the membrane, of this membrane, PLCL chitosan, nicotinamide, and green synthesized loaded nanofibrous membrane has highest activity against the antibacterial activities. Then antioxidant activity. Antioxidant activity produced strong inhibitions of free radicals by zinc oxide synthesized by green synthesis method. And then at second level, chemically synthesized zinc oxide nanoparticles and low activity by the leaf extract, that is the Osimo basilica. This is the release profile of nicotinamide from nanofibrous membranes. From both membranes, chemically synthesized nanoparticles loaded membrane as well as green synthesized nanoparticles loaded membrane. Up to 12 hours, there is the burst release of the drug from the nanofibrous membrane. It may be due to, there is the few reasons. It may be due to some drug embedded over the nanofibrous membrane, that is why that drug was fastly dissolved. After 12 hours, there is the sustained release rate was observed in both the membranes till 96 hours. As the membrane was degraded, the drug was released. The rate was the sustained release rate. Then the cytotoxicity test. Cytotoxicity test was performed over the NIH-3T3 mouse fibroblast cell lines, on which all membranes show the cell viable. 100% जो है वो successful results आए थे. All cells जो थे सब membranes के ऊपर जो है वो cell viable थे. Means के loading amount of nanoparticles within the membranes are sufficient, having no toxic effect over the cells. Then the in vivo assessments over the nanofibrous membranes in rats. We have applied these all membranes over different groups. We have made these 3 to 4 groups for particular days, 3rd day, 6th day, 9th day and 12th day. On each day, from each group, we have to take out a one rat, again open their bandage and see the condition of wound and measure the wound. So from the control up to 12th day, there is also a presence of minute wound. But in zinc oxide, green synthesized zinc oxide loaded nanoparticle as well as nicotinamide loaded membrane, on 12th day, there is the successfully recovery of completely recovery of the wound along with the hairs. Hairs भी जो है वो completely आ गए हैं और wound भी जो है वो completely यहाँ पे regenerate हो गया है. 1 kilo hair size up to 12 days. 12 days में जो successful 12th day पे जो successful rate आया है हमारे पास वो इन 2 में आया है. Zinc oxide loaded green synthesized as well as chemically synthesized nanoparticles loaded nanofibrous membranes produced 100% recovery of the wound on 12th day as compared to other membranes in which no loading of nanoparticles. This is the biopsy results. Microscope examination on 12th day. Just 12th day के जो सैंपल्स थे उनको लेकर हमने जो है वो बायोप्सी के लिए ऑब्जर्व किया था. तो बायोप्सी में जो ग्रीन सिंथेसाइज्ड लोडेड नैनो पार्टिकल्स मेम्ब्रेन्स थी उनमें कोई भी जो है वो इन्फ्लामेटरी सेल या स्कैब डेवलपमेंट नहीं था. 100% जो है वो हमारी जो एपिथेलियल लेयर थी वो डेवलप हो चुकी थी और कम्प्लीटली जो है वो वेजल्स जो है वो डेवलप हो चुकी थी एज कम्पेयर टू अदर सैंपल्स. एम अचीव्ड. हमेशा जो एम सेट होता है वही लास्ट में जो है वो अचीव करने की हर कोई कोशिश करता है. जिस तरह स्टार्टिंग में मैंने बताया था आप लोगों को कि एम एक ही होता है, उसके बेस पे ऑब्जेक्टिव्स जो है वो आपके मल्टीपल होते हैं. तो एम हमारा ये था कि हम नैनोफाइब्रस मेम्ब्रेन जो है वो डेवलप करें, सिंथेसाइज करें जिसमें बायोएक्टिव मॉलिक्यूल लोडेड हो और वो सक्सेसफुली हमने जो है वो डेवलप कर दी, सिंथेसाइज कर दी. उसके बेस पे जो हमने ये सारे ऑब्जेक्टिव्स थे वो भी अचीव कर लिए. Zinc oxide nanoparticles synthesized successfully by 2 different methods, chemical as well as green synthesis. Same analysis show a clear round shape of nanoparticles were produced, while UV visible spectroscopy showing absorbance peak at 360 nanometers and 370 nanometers. While different solutions of PLCl and chitosan loaded with nanoparticles as well as nicotinamide prepared for the synthesis of nanofibrous membrane. These 6 types of solutions are prepared. In the result, 6 types of membranes were produced. So same results produced at various fiber diameter in each membrane. Again, the zeta potential of nanoparticles in distilled water was measured. That is the minus 17.1 millivolt in chemically synthesized nanoparticles, while zeta potential in green synthesized nanoparticles, that is the minus 23. The highest radical scavenging activity was present in the green synthesized nanoparticles, up to 97% of inhibition, while chemically synthesized nanoparticles and plant extract having 94.01 and 80.901 respectively. Release rate of nicotinamide from nanofibrous membrane was burst release up to 12 hours, initial 12 hours. After 12 hours, up to 96 hours, there is the sustained release rate was observed. During antibacterial activity, highest inhibitory activity, the highest inhibitory zone was found in green synthesized nanoparticles against Staphylococcus aureus with average size that is the 13.7 and the area of inhibition that is the 147.3 mm square. While the antibacterial activity of nanofibrous membranes loaded with green synthesized nanoparticles considerably high zone of inhibition against Staphylococcus aureus is compared to nanofibrous membrane loaded with chemically synthesized nanoparticles. Cell viability. Cell viability also similar in chemically synthesized nanoparticles loaded as well as green synthesized nanoparticles loaded. Cell viability is 98% as well as 99.5%. Over the NIH 3T3 mouse fibroblast, cell lines with initial concentration of cell lines 5 into 10 raised to power 3 cells per minute. Microscopic examination of wound chloride, PLCL-chitosan, nicotinamide, and green synthesized loaded nanofibrous membrane as well as chemically synthesized loaded nanoparticles, nanofibrous scaffolds revealed a better healing properties in open wounds, 100% and 99.5%, means almost there is the similar rate, up to 12 days. H&E staining on 12th day, that is the 6 mm wound size over the skin of rats were healed within the 12 days, that is the early wound chloride, with complete epithelium, highly vascularization, collagen deposition, angiogenesis, without scab development, and also some other key parameters were significantly boosted by the green synthesized zinc oxide loaded nanofibrous membrane as compared to other nanofibrous membranes. In the light of above findings, the research project was concluded with this statement: synthesized nanofibrous membrane of PLCL-chitosan loaded with zinc oxide nanoparticles, green synthesized, and nicotinamide was very effective in early wound repair and skin regeneration, as well as show a very strong antibacterial as well as strong antioxidant activity. These are the references of my study. Thank you.
Yes, I have a bunch of questions. So my first question is, you actually need to stitch the nanoparticle, the nanofiber which has the incorporated nanoparticle of zinc oxide on the wound. Stitch it to the skin.
Nanofibrous membrane.
Yeah.
Which nanoparticles are loaded.
Yeah.
Yes.
You have to stitch it. So you actually also have to stitch on the human skin as well.
No, still not. Still, we are on initial trials.
Oh, right.
For 9 months.
All right. Also, what is like the function of the bioactive molecules? How does it actually heal the wound?
By promoting, activating the cell, those molecules which are especially involved in the healing processes. Activate करता है उन molecules को जो healing में involved होता है.
Okay.
सर मेरा क्वेश्चन जो है मैं उर्दू में पूछूंगा ताकि अच्छे से कन्वे कर सकूं. जो आपने कहा हीलिंग की बात, तो सर देखिए जब हम कोई इस तरह का पैरामीटर जज करते हैं तो हम जैसे कि आपका टारगेट जो था वो आपके लिए नैनो फाइबर्स थे, मेनली नैनो फाइबर्स. बट जो हमारे पास जो माउस होता है उसके अपनी भी एक इम्यून होता है ना, अगर उस पर कोई वूंड आ जाता है और आप नैनो फाइबर अप्लाई नहीं करें तो वो खुद भी हील होता है ना. तो मतलब इस तरह तो अगर उधर भी हीलिंग चल रही है और आप वो भी अप्लाई कर दे, उधर भी हीलिंग चल रही हो, इस तरह हम तो कंक्लूड नहीं कर सकते ना कि ये कितना होगा.
We have compared also a normal healing process in rats. Normal healing takes a time. It may be that 14 days, 18 days, 20 days, depend upon the wound. कितना day. But in our study, the wound gets recovered within the 12 days. With complete epithelialization, as well as complete hair follicles, hairs are completely regenerated.
So you want to say
that early regeneration.
So you want to say that it just increase the
process of healing.
ये जो आपने निकोटिनामाइड यूज किया था, इसका साइज भी इनकॉर्पोरेट आपने नैनो साइज से भी इनकॉर्पोरेट किया था, ये नॉर्मल ये भी
इसका जो है वो मिलीग्राम्स मतलब
इसको आपने
नैनो में कन्वर्ट नहीं किया,
जो थी उसी को आपने सस्पेंड करा,
वही हमने सॉल्यूशन में ऐड की, सॉल्यूशन में इनकॉर्पोरेट करने के बाद वो सॉल्यूशन हमने रन किया इलेक्ट्रोस्पेक्ट्रोस्कोपी, तो उसमें से फाइबर्स
तो जो आपने नैनो पार्टिकल्स इनकॉर्पोरेट किए वो भी फाइबर बनाने से पहले सॉल्यूशन में ही आपने सस्पेंड करा के उससे फिर फाइबर।
सर ये बताया कि जो नैनो पार्टिकल का जो आइडियल साइज है वो कितना होता है? अगर जो आइडियल साइज मतलब आप फैब्रिकेशन नहीं करते हैं तो क्या वो इफेक्टेड रहते हैं दवा की जो फैब्रिकेशन पावर है?
नैनो की जब हम बात करते हैं तो नैनो वर्ड से ही क्लियर है कि जो पार्टिकल्स हैं, जो मॉलिक्यूल्स हैं या जो मेम्ब्रेन्स हैं उनकी साइज क्या होनी चाहिए विद इन नैनो रेंज। नैनो मीन्स के 10⁻⁻⁹ मीन्स के 1 से लेकर 100 के दरमियान आनी चाहिए। पर अगर अबव 100 बियॉन्ड द लिमिट्स अगर जा रही है, उसके रिजल्ट्स हमें मिल रहे हैं तो फिर वो एक्सेप्टेबल है। अगर बियॉन्ड लिमिट जा रही है और रिजल्ट्स भी नहीं मिल रहे, मीन्स के देयर इज द प्रॉब्लम इन द सिंथेसिस ऑफ योर मटेरियल।
सर ये जो नैनोफाइबर्स होती है इसका डायमीटर किस तरह कंट्रोल करते हैं कि इस रेंज में ही आए?
डायमीटर कंट्रोलिंग के डिफरेंट पैरामीटर्स हैं। जब हम इलेक्ट्रो स्पैनिंग की बात कर रहे थे, इलेक्ट्रो स्पैनिंग में मैंने डिफरेंट पैरामीटर्स बताए थे। सॉल्यूशन पैरामीटर, इसमें कंसंट्रेशन थी, मॉलिक्यूलर वेट था, विस्कोसिटी था। तो उसी टाइम मैंने बताया था कि अगर हमारी प्रॉपर कंसंट्रेशन होगी तो प्रॉपर नैनो फाइबर्स बनेंगे, प्रॉपर कंसंट्रेशन नहीं होगी तो नैनो फाइबर्स नहीं बनेंगे। इसी कंसंट्रेशन के बेस पे जो है वो फाइबर डायमीटर भी बनता है। और दूसरा फैक्टर जो अफेक्ट करता है वो डिस्टेंस, किसके दरमियान? कलेक्टर के दरमियान और नीडल के दरमियान। अगर डिस्टेंस शॉर्ट होगा तो फाइबर अर्ली कलेक्ट होंगे, जब अर्ली कलेक्ट होंगे तो उनको स्ट्रेचिंग का टाइम नहीं मिलेगा। जब स्ट्रेचिंग का टाइम नहीं मिलेगा तो क्या होगा? फाइबर का डायमीटर थोड़ा बड़ा होगा। पर अगर कलेक्टर और नीडल के दरमियान डिस्टेंस ज्यादा होगा, जब डिस्टेंस ज्यादा होगा तो फाइबर को स्ट्रेचिंग का टाइम ज्यादा मिल जाएगा। जब टाइम ज्यादा मिलेगा तो फाइबर का डायमीटर जो है वो बिल्कुल माइन्यूट हो जाएगा।
Thank you so much for mashallah amazing slides. And I have many questions. First, I need to understand about the whole story here. Now you have this membrane. The try to see, okay, the try to see the Europolymer here. Okay. The zinc oxide, your nanoparticles.
Loaded into the membrane.
Okay. And the nicotine is your medicine?
Sorry?
The nicotine amide, it is your medicine?
It's
your medicine for
no, just purchase from the market and use.
For what? I went to
Nicotinamide, vitamin B also have an effective role in skin regeneration. That's why we have this.
Okay, so what's the purpose now of the zinc oxide and what the chemical reaction between them? How all these components you collect of them for your membrane? I need to know what the role of each of this component, number one. Number two, is there any chemical reaction there? Because I haven't seen any equation here about if there are any bonds.
Yeah, there is the relation. Nicotinamide as well as zinc oxide nanoparticles. The membrane we have used without nanoparticles in which nicotinamide is loaded, they have also a role in regeneration, but that is not effective as compared. When loaded again nanoparticles with nicotinamide, there is the effective role, which promotes the healing process again.
The nanoparticles you have used, it is the cross-linking or maybe...
Yeah. With when we have to mix it within the solution, definitely there is the cross-link between the molecules.
What do you think about what the type of the connection here, the chemical? Are there any chemical bonds between your...
Yeah, within the chemical characterization, I have also mentioned there is the...
I see the FTIR, it is really the same. Nothing else. And also here about the relation you said that we have releasing of nicotinamide. According to what this releasing has happened now? Is there any empiric bonding between your
Yeah, the membrane is a biocompatible and biodegradable. When the membrane is degraded, the loaded molecules are also released.
Because the membrane is degradable. Okay.
The polymers, that is the polymers. Polymers are biodegradable when touched with the skin, or even though we have to dissolve within the artificial release medium, that is the PBS. In PBS, the membrane slowly and gradually degradable. As the membrane is degraded, the loaded bioactive molecules are also released in the medium. From the released medium, we have to check it.
It's degradable after 12 days or after which day?
Sorry?
The period. After 3 days, 4 days, 6 days. Or...
Up to 96 hours, up to 3 days, we have to check. During initial 12 hours, there is a burst
3 days, we need to replace this membrane or this plaster.
No, up to 96 we have checked. But maybe beyond 96 hours, it has the activity. In next levels, we will go for that. But in our study, up to 96 hours. It has the, still 96 hours, there is the sustained release. After 96 hours, maybe there is the sustained, or maybe drop down.
Why you didn't show us any comparing between your study or the literatures?
Here I don't mention here, but in earlier my thesis
what the
I have
optimization of
already did. But here just my is a presentation is a lecture, that is why I can't relate it with literature.
I noticed some of your results you mentioned about the green synthesis give you best results.
Best results is correct.
How you can interpret these results?
Why it may be due to the some bioactive molecules from the leaf extracts.
Okay.
So having other bioactive molecules that may be boosted the healing process or antibacterial activity or antioxidant activity. Yes.
Okay. And I noticed that you, for preparing these nanoparticles, you use centrifuge under 3000 RPM. As we know, for separate the nanoparticles, we need more.
Not separate the nanoparticles.
For what is the
centrifugation performed for the जो extract था हमारा उसको जैसे separation of the extract उसमें जो extract आ जाए उसको separate करें और जो filtrate हमारे पास बचा है उसको utilize करें.
Thank you so much.
सर इसके ऊपर आपने कुछ पब्लिकेशन है जो हम
जी
उसका कहाँ पे मिल जाएगी क्या उसका गाइड
उसको जो है वो आपको नैनो पार्टिकल्स की अभी आपको मिल जाएगी. इंटरनेशनल जर्नल है, इंटरनेशनल जर्नल ऑफ केमिकल एनालिटिकल केमिस्ट्री.
सिर्फ नैनो पार्टिकल की है
अभी नैनो पार्टिकल
अच्छा इस पूरे की नहीं
पूरे की इन प्रोसेस है. वो इन प्रोसेस वो अभी रिव्यू प्रोसेस में चल रहा है वो भी बहुत बढ़िया.
Sir, you use your nanofibers before and after. Did you check the surface of these nanofibers before and after by SEM and by another techniques?
Yes.
To see the structure before and after.
Yes.
Did you mention or not mention? You mentioned?
Simply I have mentioned at the time of separation.
Not time, it's just the surface of this
Surface,
nanofiber.
it's surface, physical surface.
Before and after using this.
Physical
nanofibers.
surface.
And maybe
Physical surface like a single page, like a single white cloth.
I mean, now you take your nanofibers before using, you check, characterize. After using, you take it and check.
No, after using we can't. Because after characterization, that is degraded.
Yeah.
That is degraded. Membrane was degraded.
Why didn't you use this NMR here? Because we have this zinc oxide. Because we have oxygen here, maybe oxygen is bounding with hydrogen, and you can check the structure of this.
But हमारे पास इसमें that is facility.
You have the 2 peaks in your calculation about UV. 2 peaks for green.
For
This
green,
is
as well
just
as
for green.
One for green and other for the chemical.
Did you check the diameter for the 2 peaks?
No, diameter.
Why
Just
you
green.
have here the peaks here for green and here for chemical?
It is due to the different, maybe due to the different functional groups. Because In chemical synthesized, there is the presence of just chemicals. But in gene synthesized, we use the plant extract. In plant extract, there is also other compounds.
Did you check the thickness of your nanofibers?
No.
Thickness.
No.
Controlling the thickness of diameter and radius of this nanofibers.
Just radius of the diameter of the nanofibers membrane by using images software.
I think you have this is related to radius of your nanofibers.
No, that is the fiber diameter. That is the fiber diameter.
Well, sir, basically when we do some kind of reaction, so we look for the leftovers. Right after when the reaction is done, so we look for the byproducts. Is it toxic or not? So if when your healing process is done, so is there any kind of side effect? Like you said that the nanofiber they get dissolved, right? They go on to the skin. So do it has any kind of toxic effect?
Initial cytotoxicity test gave a 100% success results. There is no cytotoxic effect over the cells by these type of membranes. But after attention over the membranes, we can't perform. क्योंकि टेस्ट के लिए जो है वो DNA एनालिसिस और DNA के ऊपर जो है वो हमें टेस्टिंग करनी पड़ती है. उसका जो है वो प्रोसेस नेक्स्ट स्टेज है.
Sir, do you have
कौन सी जीन्स को एक्टिवेट कर रहा है, जीन्स की एक्टिवेशन हो रही है, क्या हो रहा है, क्या नहीं हो रहा है, इंटरनल मैकेनिज्म.
सर क्या आपने ये टेस्ट किया कि जब फाइबर्स लगा वो अटैच हो गया, वो स्किन का हिस्सा बना या मतलब उसकी किस तरह की मॉडलिंग? राइट, ये तो केमिकल चीज था ना, बाहर आपने बनाया था एक चीज नेचुरल है, ऑर्गेनिक है. एक बाहर से एक चीज बनाया हुआ वो ऑर्गेनिक में फ्यूज हो रहा है ना, वो मतलब उसका आपके पास कोई प्रूफ वगैरह कुछ?
जस्ट जो उसको जब हमने ऑब्जर्व किया 3rd डे 6th डे 9th और 12th डे, तो उनकी जो है वो मेम्ब्रेन के ऊपर जब हम लगाते हैं तो वोंड क्या है, ओपन वोंड होने की वजह से जो मेम्ब्रेन है वो बिल्कुल क्या हो जाती है, उस वोंड के साथ अटैच्ड हो जाती है, एब्जॉर्ब हो जाती है. पर फिजिकली व्हेन वी ऑब्जर्व इट वोंड, ओपन वोंड, वी कैन सी देयर इज द प्रेजेंस ऑफ मेम्ब्रेन. Up to initial days, 3, 6, but after 6th day, 9th and 12th day, there is no visibility of membrane. Means the membrane is completely degraded.
सर जो वुंड हील हो गया, तो उसी को अगर हम दोबारा लेके चेक कर ले और नॉर्मल स्किन के साथ कंपेयर करते हैं, उसमें कोई केमिकल चेंज वगैरह, केमिकल कंपोजिशन?
वही तो मैं कह रहा हूँ कि वो अगेन हम जो है वो जेनेटिकल एनालिसिस पे जब जाएंगे तो फिर पता लगेगा कि कौन सी जीन्स एक्टिवेट हुई है, कौन सी जीन्स डीएक्टिवेट हुई है, क्या फैक्टर्स हैं जो है वो इनहेंस में है, कौन से फैक्टर्स जो है वो रिड्यूस.
Also, as you said that you have a nanofiber which has your nanoparticles and stuff, you stitched it to the skin of the rat mice,
yes.
Right? So you checked it every day, 1 day, 2nd day, blah blah blah, so on. So each day you took off cut the stitches and removed the fiber, and then checked it, and again tested that same fiber.
Again, each time we repeat the rate. Each group has separate rates. 4 rates in each group. And for example, we take on 3rd day from the one group, one rate, we take out it and take their image, remove the membrane and leave it, discard it. Again, remaining from the remaining, on next day we remove another rate, check the effect of it.
ये जो 3 दिन बाद आपने छोड़ दिया वो नेचुरल हीलिंग पे जाएगा.
हाँ, उसको छोड़ दिया या तो उसको जो है वो बायोलॉजिकल प्रोसेस के थ्रू हम किल कर देते हैं, खत्म कर देते हैं, ताकि ऐसे ना हो कि उसमें केमिकल लगा हुआ है वो बाहर जाकर कोई हैजर्डस इफेक्ट काज करे. उसको नॉर्मली जो है वो किल किया.
आपके नैनोफाइबर में नैनो पार्टिकल की ग्रोथ तो नहीं होगी, क्योंकि नैनो पार्टिकल नॉर्मली ग्रोथ कर जाते हैं, उनका साइज बढ़ जाता है.
उनके ऊपर है नहीं, उसके अंदर है,
नैनोफाइबर के अंदर आइसोलेटेड है.
Thank you.